Now we are going to discuss certain GI hormones which are important for MCQ, right? So, main three hormones we have to read in this chapter. One is the gastrin, one is the CCK, and one is the secretin. These three hormones are very important for MCQ. Now start from gastrin. Gastrin, okay. What you have to remember here? Gastrin is produced by G cell, obviously. So G cell secrete gastrin. Where they are located? Mainly they are located at the level of anteropylori region, but main at the level of antrum of stomach. Although there are certain G cell even at the level of duodenum also, but maximum G cell at the level of antrum, right? So this is our first point. Second, when the gastrin will be secreted means stimulus. Multiple times they have asked this question. Stimulus for gastrin release. Most potent stimulus are peptides and amino acid. They are the most potent stimulus for gastrin. Apart from that, if there is distension of antrum region of the GIT, it also causes a release of gastrin. And third stimulus is calcium ion. Presence of calcium ion in stomach also produces gastrin. And fourth one is gastrin releasing peptides. It is generally produced by neuronal endings, particularly vagus nerve ending produces GRP, gastrin releasing peptides. All are the stimulus for release of gastrin. Now what the gastrin will do? Mean third, functions of gastrin. Functions of gastrin. You have to remember like this, okay? So stomach, right? This is G cell, suppose this is G cell. So G cell will produce gastrin. Gastrin first will enter inside your circulation. So I am drawing one blood vessel. So this is my blood vessels. Blood vessels. Gastrin is entering here. Through circulation, it will reach at the level of parietal cell of stomach, which are present at the level of body and fundus. Okay, so this parietal cell, this is P cell, parietal cell, located at the level of fundus and body. So how the gastrin will reach there? It will reach through circulation. Okay, we all know that this parietal cell produces H plus ion, and this gastrin is one of the stimulus of this parietal cell. Okay, so what is the main function of gastrin? This is the main function, that is stimulation of acid secretion by parietal cell is the main function. Apart from acid secretion, gastrin also stimulate another cell, that is the chip cell, which produces pepsinogen. So I am writing gastrin, the major function is acid and pepsinogen secretion. This is the main function of gastrin. Now when the H plus ion is secreted by this parietal cell, this H plus ion causes inhibition of the G cell or gastrin release. So this is a kind of negative feedback. H plus ion is most potent inhibitor of G cell or gastrin release. So whenever there is presence of acid in the stomach, it will inhibit the G cell for further release of gastrin. This is a kind of negative feedback regulation of gastrin. Now, this negative feedback mechanism will be disrupted if there is destruction of this parietal cell. If there is destruction of parietal cell, which is very common in case of pernicious anemia. Why? Because in case of pernicious anemia, in 95% cases, there is anti-parietal cell antibodies. Anti-parietal cell antibodies. So what will happen? This parietal cell will be destroyed because of antibody. Now, if the parietal cell is destroyed, then this negative feedback mechanism will not be there. If the negative feedback mechanism is not there, what will happen to gastrin release? Gastrin release will be increased. That's why it is said, in case of pernicious anemia, there is huge amount of serum gastrin is present. Okay, so pernicious anemia, there is anti-parietal cell antibody. All of you know that this parietal cell, apart from acid, it also secretes intrinsic factor, which is responsible for vitamin B12 absorptions. Okay, so obviously this pernicious anemia patient will have vitamin B12 deficiency, that will leads to anemia. Which type of anemia? Megaloblastic anemia. That's why the feature of this pernicious anemia is a megaloblastic anemia. Okay, but here we are discussing there will be increased serum level of gastrin. Main function. What is the second function of gastrin? Write down. The second function of gastrin is it increases the growth of GI mucosa, stomach and intestinal mucosa, GI mucosal surface. This kind of effect is known as trophic effect. This kind of effect is known as trophic effect, right? This is the third function of gastrin, and already we have discussed one function in previous chapter, that is relaxation of this pyloric sphincter, so that the gastric emptying will be faster in case of gastrin, right? So this is all about the gastrin, which you have to remember for your MCQ purpose, right? Second hormone. Now coming to the second hormone, that is the Cholestystokinin or CCK. Cholestystokinin, right? It is produced by eye cell of upper small intestine, that is mainly duodenum and jejunum, right? So this is produced by eye cell of the duodenum and jejunum. Also CCK are secreted by vagus neuron ending. Okay, vagus neuronal ending, it can also be secreted. So these two are the sources of CCK. Okay, now what the CCK will do? But before that, what are the stimulus for CCK? Second, what are the stimulus for CCK secretion? Most potent stimulus again is peptides. Peptides, protein digestive products, that are the most potent stimulus for CCK. Apart from that, we also know that fats are the potent stimulus for CCK, but not the most potent. So most potent, if your question, then your answer is peptides. But fat can also stimulate CCK, provided that fat are digested partially. Means if you think of intact triglyceride, it is not a stimulus for CCK. But if you digest this triglyceride molecule into free fatty acid, then they can produce CCK, right? So intact fat molecule is not a stimulus, but partially digestive product of triglyceride are stimulus. And glucose is never a stimulus for CCK production. What the CCK will do? Again we will try to draw a diagram like we have drawn earlier. So this is stomach and this is duodenum. Okay, So CCK will be produced from here, I cell. So this is the I cell, it will produce CCK. What are the stimulus? I told peptides. Now at this region the pancreas is also there. So suppose this is a pancreatic duct and this is the pancreatic acyni. So these are the pancreatic acynar cell which will produce the enzymes, and this is the pancreatic duct, right. From this I cell, when CCK is produced, it has main action on this acynar cell. That means it will secrete pancreatic enzyme. That is the main function. So number 1, CCK increases pancreatic enzyme secretion, right. So enzyme will be secreted from here, it will reach at the level of the duodenum. This enzyme will digest the food particle further, so again there will be production of peptides and other food particle, and that will stimulate the G cell again. That means more the CCK, more the digestion of the food, more digestive product, more stimulation of the I cell, more CCK production. So which kind of feedback? Positive feedback. Okay, so please remember CCK is under the control of positive feedback regulation. In case of gastrin, we have seen negative feedback regulation. Here this is positive feedback regulation. Now what are the other function of CCK? Try to understand that for the digestion of the food particle at the level of duodenum, what you require apart from digestive enzyme, you require bile. So here is your gallbladder. Okay, imagine like this, here is your gallbladder connected with this same duct, common bile duct is connected here. So CCK also causes contraction of this gallbladder. Okay, so second function I am writing, contraction of gallbladder. Any substances which produces contraction of the gallbladder are known as cholagogues. Any substances which produces gallbladder contraction are known as cholagogues. CCK is the most potent cholagogues, followed by free fatty acid, right. Similarly, you also remember another substances, similar sounding substances, which are choleretics. Any substances which increases bile secretion. Okay, any substances which increases bile secretion are choleretics. Bile salt are the most potent choleretics. Bile salt are the most potent choleretics, right. So remember this. So two function we have discussed. Now if you contract the gallbladder, obviously the bile will flow from gallbladder, but for the release of bile at the level of duodenum, you have to release one sphincter here, and that sphincter is known as sphincter of Oddi. So third function of CCK is relaxation of relaxation of sphincter of Oddi. Sphincter of Oddi, right. Not only that, you have to causes contraction of this pyloric sphincter. Otherwise what will happen? Otherwise many substances containing high acidity will reach at the level of duodenum and it will hamper the digestion process. So I have to contract this pyloric sphincter. So fourth function is contraction of pyloric sphincter, right. Number 5, increase growth of pancreas. CCK also responsible for increase the growth of pancreas, okay, tropic functions. And number 6, it augment the function of secretin. Okay, so basically CCK and secretin, they are like friend. They augment each other's action. CCK augment the action of secretin and vice versa, right? So these are all function of CCK. Last function, not the least, we have discussed that function already. CCK increases the movement of the part of GIT, okay, movement of the part of intestine, I should say. Okay, why? Because after the food particle is digested, it has to move to the distal part. CCK also causes increase movement of the intestine, right? So write down 7 function, that is increase intestinal movement. This is also done by CCK, right? So this is all about the CCK. Now coming to the next hormone, that is our secretin. Secretin. Now hormone secretin has some historical importance. Why? Because this is the first hormone which was discovered in 1902 by Sterling and Bayliss. Sterling and Bayliss. They first time discovered hormone, and that is secretin hormone. It is the first hormone to be discovered, right? Secretin is produced by S cell of upper intestine, duodenum and jejunum, right? What is the most potent stimulus? Number 2, stimulus most potent for secretin is H plus ion, that is acidity of duodenum. So acidity of duodenum is the most potent stimulus for secretin secretion. What the secretin will do? Again, you try to imagine a diagram like this. Always try to remember like this. You will remember better. Okay. So this is stomach and duodenum. So this is S cell. This is pancreatic duct, and this is pancreatic acinar. From S cell, whenever the secretin is secreted, it has main action on duct of pancreas. In case of CCK, we talked about acinar action. Now we are talking about action of secretin on duct cell, pancreatic duct. What it will produce? It will produce bicarbonate, HCO3 minus bicarbonate, and this bicarbonate will reach at the level of this duodenum. Now whenever this bicarbonate is reaching, okay, whenever the bicarbonate is reaching, it will causes neutralization of the H plus ion which is present here. What is the source of this H plus ion? H plus ion was coming from the stomach, acidic food particle. This H plus ion stimulate the S cell. S cell produce secretin. Secretin stimulate the pancreatic duct. Bicarbonate is produced. Now the bicarbonate is neutralizing the acid. Whenever you neutralize the acid, what will happen to S cell? S cell will not be stimulated, mean secretin production will decreases. So this is a kind of which type of regulation? Negative feedback regulation of secretin secretion. So for gastrin, negative feedback regulation. For CCK, positive feedback regulation. For secretin, again negative feedback regulation. So if I ask you, what is the major function that you have seen here? Major function of secretin, number 1, bicarbonate production from pancreatic duct of pancreas, right? That is the main function of secretin. Apart from that, it also decreases acid secretion from stomach. Okay, it also decreases acid secretion from stomach. Okay, right? Number 3, it augment the function of CCK. I told you already they are like friends, they will augment each other's action. So this is all about secretin, you have to remember. Apart from this major 3 hormone, you remember this hormone also, GIP. Tell me the name. GIP, gastric inhibitory peptides. Okay, this is the only hormone in GIT which is stimulated by all 3 major type of food particle, that is glucose, it is stimulated by glucose, it is stimulated by peptides, it is also stimulated by fat. No other hormone in the GIT is stimulated by all 3 types of food particles. This is the only hormone, GIP. I told gastric inhibitory peptide. Now the name of this GIP has changed. Now this is not gastric inhibitory peptide. This is now known as glucose induced insulinotropic peptides. Now this is known as glucose induced insulinotropic peptides, GIP. So again the name is GIP, but the name has changes to glucose induced insulinotropic peptide. What do you mean by that? Means it is induced by major food particle, that is the glucose, and after secretion GIP stimulate the beta cell of the pancreas for the release of insulin. Please note down, this is the normal physiological function of GIP, that is insulin secretion from pancreas. This is the only physiological function of GIP. So initially GIP name was given because it thought that GIP means gastric inhibitory peptide, it inhibit the stomach secretion. Okay, but recently it has been known that inhibition of stomach secretion, inhibition of stomach or gastric secretion is also done by this GIP, but this is at supraphysiological doses. Okay, so if you give large dose of this GIP, then this function will also be seen, but at physiological pH the only function is insulin secretion from pancreatic beta cell. Any GI peptide, if it is responsible for insulin secretion from pancreas, they will be known as incretin. Okay, so please remember this name, incretin. Any peptides from GIT which is stimulating the beta cell insulin release is incretin. So GIP is one of the incretin. GIP is one of the incretin. The most potent incretin is GLP, glucagon like peptide 1. This is the most potent incretin, but here we are talking about GIP, glucose induced insulinotropic peptides. Am I clear? Now apart from this, if you want to remember certain other hormone which are not very important for MCQ purpose, but remember peptides YY and guanylin, these 2 hormone also, if you able to remember, it's good. Both of these hormone are produced from ileum and colon. So guanylin and peptide YY both are produced from ileum and colon. Guanylin, why the name is guanylin? Because this hormone is a agonist to guanylase cyclase enzyme. Okay, GC, guanylase cyclase C, it's an agonist of this GCC, guanylase cyclase C. That's why the name is guanylin. What is the function? Guanylin increases fluid and electrolytes absorptions from intestine, right? Remember this. Guanylin increases the absorption of fluid and electrolyte from intestine. What is the function of peptides YY? Peptide YY acts at two places mainly. One is stomach and second is pancreas. Effect on two region. Stomach, what is the effect? Decrease secretion, decrease acidic secretion from stomach, and pancreas again decrease secretion. That way you try to remember, it will be good. Okay, so now look at certain MCQs. Okay, so which of the following is a primary site of production of gastrin? Very simple MCQ. Gastrin is produced by enteropyloric region of the stomach. So main is the antrum, although pyloric part also produces some of the gastrin. So this is antrum. Okay. Which causes antral gastrin release? What is the stimulus for gastrin? That is the distension, stretching. Okay, so that is the antral distension which causes antral gastrin release. Acid is an inhibitor. Calcitonin is also an inhibitor of gastrin release. Okay, motilin is secreted by which cell? We have already discussed. It is secreted by MO cell. MO cell are mainly present in duodenum and jejunum ileum. Although, so duodenum, jejunum, ileum everywhere, but main is duodenum and ileum. Although it is also present at the level of stomach. Okay, so this is all about GI hormone.